Welcome, everyone. It's a great pleasure today to welcome Professor Maurizio de Pitta. He is the principal investigator of the neuron-glial interactions lab at the Cremble Research Institute in Toronto, Canada. Welcome, Maurizio.
Good afternoon, good evening, wherever you folks are. Nice to have you here and nice to be here. Thank you, Bruno.
Perfect. So we're going to talk about glia, and sometimes you are not so clear about glia. So sometimes it's called glia cells, neuroglia, gliocytes, and those are cells of the central nervous system or peripheral nervous system that do not produce electrical impulses like the neurons. You have the macroglia and the microglia. The macro is the astrocyte. We're going to talk a lot about these cells in the shape of stars, astrocytes. The oligodendrocytes are the cells that create those myelin sheath around the axons. The cells of the ependyma, the ependymal cells around the ventricle, those are the macroglia. And the most common cells of the macroglia are the astrocyte, the most abundant cells. Then you have the microglia, small cells. They are specialized macrophages, and they will destroy and eat debris and waste product, and they're very mobile. In the peripheral nervous system, we have the Schwann cells. They're a bit like the oligodendrocytes, and you have also the satellite cells. So glia means glue. He was this German physician, Virchow. He was known as the father of modern pathology. He coined a lot of words like embolism, thrombosis, parenchyma, spina bifida, leukemia, the Virchow-Robin spaces. Maybe we'll talk about them later on in the brain. And glia for glue. We thought those cells, these glia cells, this neuroglia was just a glue around the neurons and didn't have such an important role. And recently, there's a lot of research on astrocytes, and Maurizio worked on the relation between astrocytes, the glia cells, the neurons, and the memory. So Maurizio, can you remind us the different types of memory in humans and maybe define working memory?
Sure, so with pleasure. Thank you for the extensive introduction. So when you ask me about the different types of memory, and the memory that we probably want to talk about it today in terms of where astrocytes could contribute, right? Let me give you a little bit of a big picture. So you can define memory in two directions, right? Memory as something that we use to store information, and memory as something that we use to retrieve information. Long-term memory, something that we retrieve to cherish moments and to use also to make up our own experience. But we also have another kind of memory, which is the short-term memory. So this kind of memory is something that it's more used in our daily life, right? Sometimes we have short-term memories in the sense we need to remember some information for a project, right? That we want to carry on from now to the next 1 year. And then we have a very special part of short-term memory, which is something very volatile. It's this kind of memory that is called exactly working memory. So it's a memory that we work on our daily life. And we work with also because without it, we couldn't do pretty much anything. So you are experiencing this working memory very like naively right now, listening to me. Mm-hmm. So inside your brain, you are collecting like a recipient, all the words that I'm telling you, right? To make up a story of what eventually I am telling you by my phrasing. So, and this kind of buffer, it's one of the ways this working memory works. At the end of this conversation, right, you will not remember that many details of my conversation, you will extract again an information that will become a short-term information that you will carry on in your next week, 2 weeks. And then out of that, you will consolidate that probably in something that is going to be long-term. And you will say, wow, that guy was amazing. That was a nice conversation, but you will not remember exactly all the details of this conversation. Now, working memory is something that practically we definitely use on every moment, right? So for example, let's say you want to cook potatoes, right? You have to set a timer, and after that timer, you need to drain the water. So that part of the process that you make up your mind with, you put an agenda, pinpoint, say, I have to remember myself to drain the water in 15 minutes from now. So that is an example of working memory. In 15 minutes from now, your reminder will bump up, you will drain the water, and soon after, you will not need to remember that you set a reminder to yourself. You won't go on with the next task. It would be like peeling the potatoes, or I have to remind myself to pick up the salt after that, and so on. Every time you refresh this memory fast enough, right, to create a new task, to go on the next task. So these are the separations of memories in terms of storing information.
So Maurizio, the working memory, is it mainly in the prefrontal cortex in primate, in human, or you have other areas for it?
No, so that's a good point. Originally, right, working memory, so this, how do we know about working memory, or at least what we call the cellular correlates of working memory? Because back in the 80s, okay, we figured out that this idea of storing something in memory, something in our brain temporarily, which on the neuronal level, as you mentioned before, right, is something that we can record on the electrical signals. So ideally and intuitively, right, for us, this storing is shown as, okay, the neurons that do this kind of storing are active in that period. As soon as I give my input, as soon as I set up the clock in 50 minutes to go and turn off the potato, the fire for the potatoes. So, and this is what they observed, not in humans back in the days, mostly monkeys with not potatoes, but something similar to potatoes, images in that case. And so they found actually back in the monkeys that this, what is called persistent activation, this persistent electrical activity developed by these neurons during this period from the task setting and the actual task execution. Then after the task, the activity vanishes and they go back to normal. So was developed, for example, in the inferior temporal cortex, right, which is this area below. Then, as you mentioned, there have been many other experiments. So we have different areas, dorsolateral prefrontal, for example, then medial temporal lobe, medial frontal cortex, prefrontal cortex, and so on. So
let me show that there. So this is a brain, and you have in front of the main fissure here, you have the frontal lobe. So this is the frontal lobe all the way here. This is parietal lobe in the back. And you have, as you know, you have the motor pathways right in front. In front of those motor pathways, you have the prefrontal cortex. And then in the very, you are dorsolateral, for example, here, the ventrolateral. So dorsolateral prefrontal cortex is going to be here, correct? Somewhere here is going to be your middle prefrontal cortex. So dorsolateral is going to be on each side here, something like that. So here you're going to have your working memory. You're going to have also, you said the middle temporal?
Inferior temporal and medial temporal.
So inferior temporal, middle temporal. What else you said? So prefrontal cortex.
Oh, you have well,
Parietal?
inferior temporal, medial temporal, you have also parietal lobe sometimes. The main idea here is that you don't have just an area that encode for the working memory. You have a distributed network made by different areas that together they carry on different things in a unified synergistic fashion.
They are all active
So,
at the same time, or some region?
no, depends. They are not necessarily active all the time, but the point is, or they can be, depending on what task you are doing.
So, Maurizio, this dorsolateral prefrontal cortex, sometimes they say it's one of the most recent parts of the human brain. So that's something that more specific to primate and human, or make it a little bit more sophisticated type of working memory, or?
That's a nice question. I don't have enough knowledge to tell you that probably it's mostly humans. I can tell you that not necessarily it's only humans. So there are like famous videos on YouTube that shows that, for example, the buffering or the capacity of working memory. So how many items, how many things I can store in my working memory before releasing, right? And so one of these typical videos is putting a human and a monkey in front of a screen, and the screen shows for a very little second lots of numbers, okay, in different areas of the screen, and the task is after 1 second or even less than a second, these numbers disappear. They only remains a square, and the subject, monkey or human, is asked to touch the dots on the screen in the order of the numbers that they represented. And so there are many, so about like 7, 10, 15 of these dots. You clearly see that the human subject barely remember the first 2 or 3 dots and is not able to complete the task. The monkey, surprisingly, within not really like even probably 5 seconds, is able to touch all the dots and go to the next task. It's fairly stunning. There is no real knowledge to what I know so far that working memory could be found, for example, in invertebrates or like other organisms, right? Although I wouldn't be surprised they could find it in the octopus. I mean, octopi are fantastically fascinating.
Perfect. So let's go into astrocytes. In human, you have a lot of astrocytes. If I used to hear that you have about 4, 5, 6, 7 astrocytes for each neuron.
So this is a misconception, to be honest. So, but we can. I'm going to elaborate on that
because
shortly.
I hear different things about that.
Yeah. So it's a misconception. So for 2 reasons. Okay. Historically, I can give you the anecdotal version. So one of the selling points back in the days about the importance of glia, because I mean, till 20 years ago, glia were still considered not much more than a gluing thing. So I prefer the Greek pronunciation, glia, right? Because glia, it comes from the real word, the Greek word, glia, which literally means glue. But I mean, you can pronounce it with the English pronunciation, so it's glia. Now, as I said, up to 20 years ago, there was not so much because the technology was not there, right? So all we know nowadays and all this big fuss about glia is because by the end of the 90s, by the end of the 80s, we have this introduction in neuroscience and experimental neuroscience of fluorescent dyes, right? So we are able essentially to measure for the first time quantities that are not strictly electrical. So quantities such as ion concentrations or species concentrations. And the way we are able to do that is because we are able to stain something that before we couldn't, right, by some kind of dyes, we call them, that goes inside the cells, and they light up when that specific reagent or something that is there is present. So dyes just light up.
So measure
So we are able.
chemical activity and not the electrical.
Correct.
So when we
Yes,
get chemical activity, we could measure the astrocyte and realize how important they are.
Exactly. So this is what happened over there, right? So they figured out that the astrocytes are electrically passive because they don't produce these sparks like a neuron do. But they are excitable nevertheless when we look at some chemicals inside. And the chemicals here can be, for example, calcium. Same kind of object, molecule, ion specifically, that you have in your bones, right? But in your bones is in a solid, or like it's chelated. It stays there, it creates this calcium oxide that makes your bone very rigid. Outside your bones, calcium, when it's a free ion, it's a very dangerous object. I mean, because it bears two charges. So ions that bears two charges in chemistry are definitely stronger than ions that bears only one charge in terms of the electrical field, right? Two charges. I mean, intuitively, you can think about that.
If I remember, the astrocyte have a intercellular calcium wave. That's the way they communicate with each other very far, and that's powerful. It's like a tsunami of depolarization or information that happen. And
yes, that's one of the theories that is behind. So however, this kind of analysis, this kind of analogy, right, it's very nice to think of this kind of calcium that propagates and create patterns. So it can somehow, in addition with the electrical channel, right, create another way of carrying or information or encoding knowledge. But again, so this idea, just to clarify on the question. So if you look at how many astrocytes you have in your brain, so generally, if you take all your brain, imagine to just break it down in terms of components and count all different cells, you come up actually with a 1-to-1 on average. So one neuron, one astrocyte, more or less, in total.
Yeah, that's what. Yeah.
The point is that it's surprising because then when you look instead at the region, right, content specific, so in the cortex, actually, you do have less astrocytes with respect to neurons. More or less, the techniques nowadays tell you that you have about, say, 4 neurons per 3 astrocytes. So one neuron every 0.8 astrocytes, something like that. In the cerebellum, it's actually 10 astrocytes per neuron.
Okay.
So it's very interesting, right? And now the question that you could ask is, why is that so? There are many possible explanations for these. One is barely on the size of the cells. In the cerebellum, neurons are much smaller than cortical neurons, right? So you might have like granule cells in the cerebellum are very small.
Yes.
And the cerebellum contains actually an enormous amount of cells, right, with respect to the cortex. It's like almost 1/10, like 9/10 of the cells in your brains are inside the cerebellum.
Because the granular cells are very, very small. They are the most abundant cells in the brain, in the central nervous system.
On the other hand, it's also possible that, and this is something that we are appreciating right now. Now, if you look at it's called the cytoarchitecture, so how these cells look like. Well, the cells in the cerebellum, the astrocytes in the cerebellum are very different with respect to the astrocytes in the actual cortex. One of the components of the cerebellum are these famous Purkinje cells, right, which are the most branched ramified neurons that you find. The giant trees, right? You can think about this kind of enormous sequoias, right, that with a lots of branching, beautiful, or like this kind of beautiful tree, exactly. But as you
All
probably
parallel.
say, they are in parallel. They create palisades. So they are giant trees when you see them like this, but you rotate them, they are bidimensional cells almost. So all the tree stays on one plane. And so actually it turns out that the majority or the most abundant astrocytes in the cerebellum are actually in between these trees. So they literally create a sort of like intercommunicating zip, right? And so they are themselves almost bidimensional in some way. You look into the brain instead, they are not any longer bidimensional. They create blobs, they create clouds, right?
Why don't you tell us
Inside...
the types of astrocyte in the brain? Because I know
Sure.
types. How many?
Yes, so I would like to tell you about it, but there are not so many types of astrocytes that I know. The majority, the distinction right now that you can think of is in the gray matter, you have what are called protoplasmic astrocytes. And in the white matter, you still have astrocytes, but they are a little bit more bushy and they look a little bit different, not so much branched. And in fact, they are called fibrous astrocytes.
So let's recap.
Now,
so fibrous astrocyte in the white matter. We may talk about them later because we have those end foot of the astrocyte on the outside of the capillary walls. And then you have the protoplasmic astrocyte in gray matter. They have a lot of organelles. I mean, in embryo, we used to have the radial astrocyte. I don't know if they still talk about those.
You do have the radial, yes, so I didn't mention. You do also have the radial. They are not called radial astrocytes because they are called more like radial glia. So this kind of radial glia, it's in terms of development, a stage that it's before the astrocyte itself. All astrocytes in the brain comes from a radial glia. And some radial glia is still there, and it's actually one of the cells that is responsible for the generation of new cells in the adult brain, okay?
No,
and
those,
yeah.
They call it the Gemuri positive astrocyte. That's kind of what is that exactly in the hypothalamus? The one that regulates the glucose consumptions. Gemuri.
Yeah, so this kind of astrocytes in the hypothalamus, so those are actually specific still in between astrocytes and radial glia. Hypothalamus, it's also a relatively less studied structure for the composition of the astrocytes. And then you have, as you mentioned before, the ependymal cells and the so-called thanacytes. So these are like specialized glial cells, right? It means that they come genealogically, like genetically from still radial glial cells, but they are not looking like astrocytes or like fibrous or protoplasmic or oligodendrocytes. They are, because they have specialized functions. So ependymals, for example, are ciliated cells, right? They are cilia, and these cilia ajar the walls of the cisternes, right, where the cerebrovascular, the cerebrospinal fluid actually goes or is created, and they also contribute to the generation of this cerebrospinal fluid.
Hey, one more thing, Maurizio. I remember a while ago I read that a single astrocyte could interact with up to 2 million synapses at a time. It was
yes. Correct. This was a very interesting study that was done back in the days, about 10 years ago, by the same group, actually, that 10 years ago also started with this idea of the glymphatic system, right? And back in the days, the experiment was very, very interesting, funny experiment. We have mice, right? And so let's imagine that we take a human astrocytes and let's put these human astrocytes inside the mice. What happens? Is the mice gonna get better? It's gonna die? It's gonna do stuff? The mice didn't die, and actually when they were tested for memory, they got smarter. So that was a very nice selling point to say, okay, so maybe astrocytes make people smarter, or like they contribute to intelligence. And so obviously it's a little bit naive, but yes, so this fact that the recognition that was one of the actual driving forces behind this revolution, and also our study, for example, that when we look at how these astrocytes fill in the space of the brain tissue, and the way they fill in, it's they not only fill in the holes, the sustaining, of course, the neuronal pathways and circuits, but they go there sustaining them exactly in the point of contacts of the neurons, which are the synapses. Well, so this fact, right, that you have something there that sustain, now that we have the lenses, meaning the technology, the microscopes, and also the tools to see how this sustaining function occurs, and it's not only mechanical, it's again chemical as well, made realize people before us and now the general community that the way synapses transmit neuronal signals, right, is modulated by a chemical signal coming from the astrocyte compartment, meaning that the interaction between the astrocytes with the neurons, right, which eventually we read out at the level of the signal transmission at the synapses, is in a way synergistic. So everything that goes on probably at the level of processing information, storing and retrieving, in particular, for example, your memories, somehow involves not only neurons, but a combined neuron astrocyte glial component.
Maurizio, could you say that the neurons and electrical signal, like your TV, you get the picture, and the astrocyte at the button to modulate the color?
Yes.
That will work.
Yes, these astrocytes that sits on our synapses, they modulate the signal according to what is the content of the signal, and they are able essentially to shift the features of our transmitter and the one of the receiver in order to, at the time, choose which features get or transmit, which is fairly amazing. It's like this kind of knob that you have on the radio, and you just tune in on whatever you want to listen to or
and doing
about.
That maybe up to 2 million neurons at the same time.
Yeah.
Amazing.
Yes. So more. I mean, I would say even more, because so 2 million synapses, of course. And the question is, these 2 million synapses, at least, right? Do they belong to the same circuit? Because if they were belonging to the same circuit, you could say still remarkable, but still I am within the circuit. The point is that very likely these synapses do not belong to the same circuit. They belong to different parts of much larger networks, right? So you are definitely doing stuff. You created this kind of islands of supervision of your local activity, where you combine, by means of this astrocyte signaling, information that comes locally as well as information that comes remotely, right? And you do very interesting things. So you can do very interesting things. So of course, this is very still speculative. In the working memory context, okay, the idea has been the following. So it's something of this type. So the astrocyte is able to modify for some time the way the synapses transmit signals. The modification here is very simple. Essentially, you can think of it in this way. The synapse transmits something. The information, that signal that is transmitted, activates the astrocytes, and the astrocyte feeds back on the synapse, sustaining that signal.
It could sustain or increase the synaptic rhythm.
Yes. So it just keep it higher. Now, it's a very simple process. You just have this kind of called positive feedback, right? I feel this signal and I feed back and say, yeah, keep it up. Now, the crucial point is that this keeping up signal or this keeping up instruction is such that the keeping up itself, right, can last longer the actual stimulus. So when you don't have any longer, for example, the picture that you have to keep in mind, still, because the astrocyte is able to maintain this information to the synapse, right? The synapse itself keeps a memory or like some kind of ghost of what was the stimulus before. And now at the level of individual cells, as well as at the level of a network, this kind of process essentially can propagate and scale up, creating this kind of activity persistent ways, okay, of encoding a stimulus after it has been presented. So you could have neurons that still are active because of this keep up signal from the astrocytes, despite the fact that you don't stimulate those neurons with the original image that you wanted to save. You see around YouTube and everywhere always this kind of beautiful artistic views of brain circuits in which the neurons and the synapses are floating in some kind of vacuum, which we believe it's extracellular space, and you see the synapses void of everything around, and you see the beautiful electrical signals going around. But it's very far from being there. Like there is no vacuum. There is no such a thing inside the brain. Everything is filled. And an essential component of this filling are actually the astrocytes and the glia. And the fact that you have a filling there, again, so it's not only just a structure, gives structure and shape. It also means that it's there for interacting. And so the interactions now taking this additional dimension of how glia, astrocytes, fill in and modify this space. You have now multiple ways that these astrocytes can modify the environment, because you are now taking into account interactions between the astrocyte and the synapses, interaction between synapses and astrocytes, so the way around, interaction between astrocytes and other astrocytes, interactions between astrocytes and neurons, and vice versa. So each one of these, it's two-way, right? So we have already 6 components, 6 different types of interactions. And then there is this other part in which you may interact with the astrocytes and all the other kind of cells. So the blood vessels, the inflammatory competent cells, so, macrophages and inflammatory response, fibroblasts, and so on. So all this kind of whole enormous world, right, of possible interaction just by the addition of one additional component leads really to fascinating ideas in more of how we work.
So the astrocyte has this wonderful neuromodulation effect. Could you call them neuroendocrine cells? You could say that's
yes, absolutely, yes. Yes. It's a way of thinking. They actually release, so these folks, they actually release common molecules that neurons release. The same molecules that neurons use to transmit signals, they are also used by astrocytes. They release exactly the same molecules.
Glutamate,
GABA, for example, and then other substances for natriuretic peptide, right? Then other things, purines, and then dicerine, and then anything. So like, a lot of neurotransmitters.
So we also have those tiny cells. We mentioned them, the microglia. I think they are more purinergic. They're just releasing more purine.
Correct.
They move a lot. Can they also come and help?
The microglia, yes, the microglia can be there. In fact, actually, you mentioned before the so-called tripartite synapse, right? The recognition that the astrocytes can modify this transmission, make them these active additional elements. So the synapse becomes tripartite, three parts, pre, post, and the astrocyte element. And then this concept has been naturally evolved, saying, no, now synapses can be even tetrapartite. Means there is a fourth component, which is actually the microglia component. So because there is a microglia there that does things that might modify the message, bringing in an interesting thing, right? Because microglia are these, so microglia genetically very distinct from the actual macroglia. So they generated during development from the yolk sac. So they have nothing to do with the actual glia as we know, the oligodendrocytes and the astrocytes. They are immune cells. They are the only innate immune cells of our brain. And still, it's interesting to think that, you know, and it's interesting, and at some point it looks like finding out Columbo's egg in some way, right? Because you have an immune cell that is sitting on the synapses, the point of contact where you are sending information around in the network. Why should I have an immune cell over there, right? And then you ask, well, how do we communicate with our immune system? How does our immune system know what is going on inside, right, our cell? And so this links to a lot of possibilities, right? Because having an immune system that is body-wide, that has a point of contact with something that is localized within our skull, right, allows potential ways of communicating from within the skull to instead our body. And so this, of course, brings in all these kind of nice theories about the gut-brain communication, the systemic mind-body synergy, and so on.
The astrocyte can be activated. I remember they used to call it A1 and A2.
Mm-hmm.
It can be really toxic. The A2, they used to call it the bad astrocyte, become to be toxic and destroy, you know, respond to many kind of substance and destroy some tissue in the brain.
So this is a good point, separating between A1 and A2 astrocytes, as well as between microglia M1 and M2 type cells, right? Bad or good ones. Instead of a more like unifying vision, saying that depending on the context, right, the cells, these two cells, essentially can start a process of differentiation in which they either gain or lose some of their functions. What you gain might be a loss from the other person, right? Somebody is giving you something. What you lose, it's a gain potentially for the other person. So there is not something like bad or good astrocytes there in pathology. You definitely want, as much as possible, when you have the tools, to look at the pathology itself in a more comprehensive fashion, put it a context, right?
Do they already coin the name astrocytopathy?
Astrocytopathy, yes,
Yeah,
the idea, yeah.
So the pathologist, so for example, could you talk about astrocyte and schizophrenia? They were talking about that. Is there something?
Yes. So it's so the astrocytopathy is this idea essentially that pathologies that we regarded for years like only as coming from neural deficits, schizophrenia, autism, even epilepsy, right? That traditionally are pathologies. Depression as well. So this neurological, I was thinking about neurodegenerative disorders, but let's talk about neurological pathologies. So neurological deficits, cognitive deficits, right? So these pathologies are traditionally regarded as a change somehow in the way neurons behave, right? Or the activity of these neurons. Because we believe there is alteration of the chemical signaling. Now, in all this kind of framework, we only think about chemical signaling coming from neurons. So we modify the chemical signaling from neurons in order to potentially mitigate or stop the symptoms. Now, with the recognition now that this chemical signaling can be modulated by the astrocytes, right? We open up a couple of perspectives there that are useful for the therapy. Not only because we, at the level of basic knowledge, we say, hey, look, these signals does not come only from neurons, but is the outcome of the interaction of these two things. The other aspect, I think, which is crucial, is knowing that there is another component in the game. It gives you, in principle, an additional door to access the pathology, to now find a way to treat the pathology. And so this is stunning. And this is why many researchers from both medical setting and the fundamental neuroscience are actually leveraging this idea that neuronal pathologies or neurological disorders could actually be, the word doesn't play well, but astrological disorders, right? Doesn't really play well in this case, but you got the idea. They could actually have an astro component, right? An astrocytic or a glial component.
Maurizio, it's an amazing discussion we had. I'm so glad you know so much, and I got enlightened talking about this different structure in the brain. And we'll be happy to read you in the future with all the results of these amazing studies you are doing. Thank you.
Thank you very much for having me here.